Hello and welcome to the screencast on Chapter 16, Section 4, where we are going to talk about the accessory structures of the eye. So we will start off with the external structures here, like our eyebrows. These are in place to give us shade and also to inhibit sweat from dripping down our forehead and into our eye. We know that sweat has water and salt in it, so that would sting, of course. Next, we have our eyelids, and the Latin term we use for this is palpebrae. Palpebrae is plural, palpebra is singular, and this is along with our conjunctiva. I believe I have a slide coming up about conjunctiva, but basically this is the opening of your eye here. That would be your conjunctiva. So let's talk about I forgot I had everything color coordinated, so let me click along here. Okay, so first we have the palpebrae fissure. We have that serving as the opening between the upper eyelid and the lower eyelid. Then we have the canthi, which you see highlighted in the gray there, and these are going to be found both medially and laterally, and it's basically where the eyelids meet one another, so that point right in there. And then we've got our medial canthus, and our medial canthus has a caruncle, and in this caruncle the modified sweat glands and sebaceous glands are going to work to make sure the eye stays nice and moist. We are going to have 5 layers of tissues, including dense connective tissue in the tarsal plate. And let me give you a better view of this tarsal plate. So here we have kind of sliced the eye down the center here, and we are looking at it from a sagittal view. So you can see here in the eyelid, the upper eyelid, we have this tarsal plate here, and that helps to maintain the shape of the eyelid. We also have a tarsal plate in the lower eyelid, just so we know it's in both boundaries. All right, when we take a look at that eyelid once again, we are also going to see eyelashes sticking out, and these can be double or even triple rows of hairs. This, of course, is to keep debris out of your eye. There's our tarsal gland. And here is our ciliary glands. So these are 2 different sets of glands. These ciliary glands are modified sweat glands and empty onto the hair follicle, and that carries it on up. And then we've got our tarsal glands, or these are known as meibomian glands as well. And these are found at the inner margins and produce sebum. So they are kind of like a sebaceous gland here. Now let's talk about that conjunctiva that I mentioned earlier. We can divide the conjunctiva into 2 different parts, and really this is just a transparent membrane that is within the opening of your eye, so all of your eye that we can see. First we have your palpebral conjunctiva. Remember that word palpebral refers to the eyelid, and that's why we just see it in the rim in that area. Whereas the bulbar conjunctiva, that is going to be everywhere else other than what we will soon know to be the cornea of the eye here. So all of this green space is the bulbar conjunctiva. Now you may have heard of people having inflammation to their conjunctiva, and this results in conjunctivitis, also known as pink eye, which you see pictured over here, where we have a reddening of the patient's eye, showing us that it has been infected. And here is another figure just showing you some of the external features that we just talked about. The medial palpebral commissure, our canthi. We've got the lacrimal caruncle here. The palpebral fissure is the opening of the eye all along here. And we talked about the palpebrae. So moving on from there, another figure here illustrating those structures for you. And more. So we've got the tarsal plate that we weren't able to see beforehand, and also we have some labels defining the conjunctiva for us. And like I said, this is the cornea, so that does not include the conjunctiva, but we could see the rest of the conjunctiva here. Okay, the other thing I want to talk about is how tears are produced and how they're secreted. So our lacrimal gland, which is found in the upper margin more laterally to the eye, is going to produce tears, and those tears are going to be secreted through these excretory ducts, or sometimes they're called lacrimal ducts, like we see here. And what those ducts lead to is to the actual eye, where it's going to wash over the eye and enter the lacrimal canaliculi. Now remember, canaliculi is just a word saying small canal. And that is going to move through these tiny openings known as the puncta. And the tears will then move through the canaliculi into the lacrimal sac, which will then descend down the nasolacrimal duct, bringing it into the nasal cavity. This is why we see a lot of people, when they're crying, they start to sniffle, is because we are overproducing these tears and they are all moving into the nasal cavity, and we have to sniff in order to pull that fluid back up. All right, now let's move into the actual eye structure here. So I mentioned in class that we have 3 tunics to the eye. We've got the outermost, which is known as our fibrous tunic or fibrous layer. And this is made up of the sclera, which is the white part of your eye, so all of this in here. And it's also made up of the cornea, which is this transparent portion of it over here. So when you're looking at a person's color of their eyes, the color is going to be in the iris, but you're looking through a window in order to see that iris, and that window is the cornea. The middle layer is the vascular layer here, the vascular tunic. This is going to include our choroid, our capillary body, and our iris. More on that in a bit. And then we've got the innermost is the nervous layer, or the retina, as a lot of people know, where we're going to find our photoreceptors. Here is a figure of a cadaveric eye, and we can see that on that sclera we're going to have some muscles attaching, and we have the optic nerve extending out. This is going to take all of the sensory input that the retina is receiving and transmit it on back, and we'll talk about that pathway in another screencast video. So I also wanted to show you this. So in this view, we have cut away the skull cap. We've pulled the brain away, but left the optic nerves, and obviously you could see the eyes here. So I wanted to leave this here to let you know that we've got our visual axis here, whatever is in front of us, and that information is moving straight back and is focused on this area known as the fovea centralis. This is almost like a little dip within the retina here, and this is where vision is going to be the most acute. We will have our cones, which are photoreceptors, really populated within this area. Now notice it doesn't head on straight back over here where the optic nerve is, and that's because this is known as our blind spot. There are no rods or cones in this area where the optic nerve attaches, so that's why we kind of have it off to the side over there. And this is a figure that we will continue to come back to. It really does a good job of displaying all the different structures of the eye. We will reference this in the upcoming slides. So let's start out with the fibrous tunic or fibrous layer of the eye. Like I said, that's made up of the sclera and the cornea. So the white part here is going to be the sclera, and actually I highlight it here in the gray, and this is going to help to maintain the shape and protects our internal structures of the eye. Like in the cadaveric photo we saw, it also provides muscle attachment and will be continuous with the cornea anteriorly. Now the tissue that makes up the sclera is a dense collagenous connective tissue with elastic fibers. And our collagen fibers are large and opaque here. So we're not letting any light that's entering the eye scatter out through the sclera, even though we would have the choroid layer prevent that too. But anyway, it's just an added layer of protection, I guess. Then we have our cornea here. Like I said, this is kind of our window to the eye, and this is made up of connective tissue as well, containing collagen, elastic fibers, and proteoglycans. Now we do have a layer of stratified squamous epithelium on the outer surface, and here we're going to see that the collagen fibers are smaller, which allows that transparency to take place here. It's also avascular, which is why we're able to do cornea transplants because there's no blood supply that we have to worry about. And this will allow the light to enter through and shine onto that fovea centralis. But as it's moving through, each component, like the lens, some of the fluid in here, it might be bending the light and refracting the light in order to focus it onto the right area. Now let's move into our middle layer known as the vascular tunic. That's going to consist of our choroid. We've got a ciliary body and an iris. We'll start off discussing the choroid region, and this is going to be the posterior portion of the uvea, or this middle layer. This has all of the blood vessels here for the eyeball, so very vascular. It also has a brown pigment to absorb any light moving through, so we prevent any scattering of the light, and it prevents visual confusion. Now we also have this ciliary body here. Inside this body, we will have a ciliary muscle that will then attach to the suspensory ligaments, allowing us to change shape of the lens. And lastly, we have the iris here. Like I said, this gives the pigment to the eye and is a muscle. Now I wanted to show you this histology view of the choroid plexus. You can see in here that brown pigment that is going to absorb the light. Moving into some more detail about the ciliary body, like I said, we are going to contain a ciliary muscle in here. Now, in this slice through the eye, you're only seeing one perspective, but I want you to understand that this muscle rotates throughout the entire eye. So kind of like the iris, it's going to completely surround and be like a circle around the iris. Okay, so those ciliary muscles, like I mentioned before, are going to attach to these suspensory ligaments. They're also called ciliary zonules that will then place tension on the lens, changing its shape so that we can focus light in the correct area. And we are to the third structure that we mentioned, which is the iris. This is the colored part of our eye, and it regulates the amount of light that is going to move through into the eye and onto our visual receptors, the photoreceptors. And what allows that light to move in is the pupil. This is just the opening, and it too is helping to regulate the amount of light entering the eye. So I really like this figure here because it really shows you the two muscles within the iris. So for close vision or bright vision, which you see over here, we are going to utilize our sphincter pupillae muscle, or the circular muscles. They will contract, which will then allow the pupil to constrict, lessening the amount of light that's going to enter the eye. When we are in dark light or in dim light, or even if we're looking at something at a distance, we are going to use our dilator papillae muscles, or our radial muscles, to contract, allowing our pupil to dilate and take in more light. We also know that our pupils change based on our emotional state. So we are If we are seeing something appealing or something that requires problem-solving skills, then our pupils will actually dilate. And here's just another figure depicting what we just discussed. So in bright light, we see constriction of the pupil using the sphincter papillae muscle. Within low light, we will have the pupil dilate using our dilator pupillae muscles. And I didn't mention that it's our parasympathetic nervous system that allows us to constrict, whereas it's our sympathetic innervation that allows us to dilate. All right, so that takes us to this slide where we are going to talk about the retina. So the retina is made up of two layers. We have the pigmented retina, in which you can see over here, and this is going to be pigmented simple cuboidal epithelium, and the pigment of this layer and choroid help to separate the sensory cells and reduce the light from scattering out. And the reason it is pigmented is because these little spots here are representing the melanin. Then we have our sensory portion of the retina, and this is the inner layer of rod and cone cells that are sensitive to light. So let's take a look at some of the cells here. We have our photoreceptors lined up over here. These are going to transduce the light energy. And then we have the cells that transmit and process the signals. So we have our bipolar cells here in the pink. We have our ganglion cells that are in the white, and our amacrine cells in this light teal, and our horizontal cells in the dark green. And they're all communicating to get that sensory information from our photoreceptors over to eventually what will be a ton of axons that will form the optic nerve. So let's talk about those photoreceptors a little bit more. We have our rods, which are a type of photoreceptor, and they are going to be the most numerous at the periphery region of the retina, away from an area known as the macula lutea. So you can see here our cones are going to be the yellow ones, we'll talk about those in a minute, and the purple are representing the rods. These are our photoreceptors. So what is this macula lutea? So you can see this is a picture of the retina when you go to the eye doctor and they dilate your pupils, or if you use one of those cool machines that basically scan your retina, you can see that this darkened redded area is the macula lutea. And this is where we're going to have more of the cones located, whereas the rods will be around the periphery over here. And these rods are going to operate in dim light, so they're best for black and white vision. And if it's damaged, there will be disturbances within our vision that will lead to night blindness. It may provide indistinct, fuzzy, non-color peripheral vision. And here's just another view of the macula lutea and where it exactly exists. So notice here is going to be our optic nerve, and we'll soon talk about the blind spot. So it's a little off to the side over there. And here's a nice little close-up. So it's this area right in here, kind of looks like a donut. And in the center of the donut is going to be an area known as fovea centralis. So we'll talk about that in just a moment. And just showing you another image of the rod and the cone. Don't worry about this physiology here. Moving into our cones, this is found in that macula lutea that we discussed so much, and we can now talk about the fovea centralis. So there is our macula lutea once again, and in the center of that donut is going to be the fovea centralis, and this is the area where we have the greatest visual acuity. So we'll have our photoreceptor cells tightly packed in that area. And here's another close-up. Macula lutea in the green and the fovea centralis in the center. So these cones are going to help us operate in bright light, and they provide high acuity color vision. When we see a certain color, for instance, this orange that is colored orange, all of the light is being absorbed by this orange, except for the orange color that is being experienced. So it reflects off, and that is the color that we are experiencing. For instance, if we were looking at a banana, all of those colors are being absorbed into the banana except for the yellow color, which is reflected out and into our eye. Now let's talk about that optic disc that I mentioned. So as the light passes through the eye, it moves through the lens, and the lens's job is to focus that light here on our fovea centralis. Now, if you look off to the side, you see this bright area that looks like a sun. This is the optic disc and is also known as our blind spot. This is an area where the blood vessels are going to enter the eye, and also where nerve processes from that sensory retina are going to meet to exit the eye. And therefore, we're going to have no photoreceptors found in this area. Now let's discuss some of the internal chambers and fluids of the eye. If we look at the posterior segment of the eye, which is going to consist of the area posterior to the lens, so all of this area in here, we will contain a vitreous humor. And that is going to transmit the light, so moving through the lens, to that fixed area of the fovea centralis. We're going to support the posterior surface of the lens in this chamber, which contains the vitreous humor. Humor, by the way, is a term that we can use for a fluid, even in this circumstance when your vitreous humor is more gel-like. And also, we are going to hold the neural retina firmly against that pigmented layer, the choroid. And will also contribute to intraocular pressure. So sometimes when somebody has glaucoma, they get pressure built up in the anterior chamber of the eye, which then puts pressure on the posterior segment of the eye, and that is going to then impinge upon some of the photoreceptors, and we will lose some of the peripheral vision that we have in glaucoma. All right, now let's talk about the anterior segment. So that's all of this area anterior to the lens. And this anterior segment can be broken into an anterior chamber and a posterior chamber. So anterior chamber is anterior to the iris, so between the cornea and the iris. And the posterior chamber is found between the iris, so this is our iris, so we're clear, and the lens itself. This area is going to help maintain intraocular pressure. We do have these special drainage areas in which we are going to have the aqueous humor secreted by the ciliary processes here into the posterior chamber. And then that aqueous humor will move from this posterior chamber to the anterior chamber through the pupil. Now, excess aqueous humor is going to be reabsorbed by some veins here known as the scleral venous sinus. And when this area gets plugged up, that's when glaucoma can take place. Let's talk about the lens a bit more now. This lens can change shape in order to focus the light on the retina. We have cells within it that have lost organelles and are filled with a crystalline protein. The shape of the lens will determine the degree in which the light is refracted or bent, and the shape is going to be determined by our ciliary muscles that lie here in the ciliary body and the suspensory ligaments, or those zonule ligaments. So that arrow is representing the light, let's say. And here's another illustration here demonstrating far vision and near vision. So obviously here we are looking at some mountains, and you can take a look at the lens, how it has flattened out and the ciliary muscles are nice and relaxed, keeping the suspensory ligaments taut so we can see that mountain range in the distance. Now, if you were to look at the time on your watch, the lens is going to have this thickened, more spherical or fat look to it. And it does this because the ciliary muscles are contracting, moving that ciliary body closer to the lens itself, and that causes the suspensory ligaments to relax. And just a clinical moment here on cataracts. This is when we have some small opacities within the lens, and usually it's going to be a result of aging. So typically this happens right around the age of 65 or older. And it's difficult to focus on close objects because of that clouding, and so we will have reduced visual clarity and color intensity. So what needs to happen is the lens needs to be removed if it is starting to interfere with normal activities, and there are new surgical techniques that include phagoemulsification. The opaque center of the lens is going to be fragmented using ultrasonic waves, and I have an image that will show you this. There we go. So here's the eye with the dense cataract, and then that cataract is broken up. In this instance, it is going to use ultrasonic waves in order to do that, and then we can remove it and fold up a artificial lens to place over here where the cataract was. And now we have a brand new clear lens in place. Pretty amazing. All right, now let's go through some clinical terms. So ametropia is going to be when we have normal vision, meaning that we have parallel light rays that are focused on the retina. Now, for those of you that are farsighted, that means your eyeball is too short. So when these light rays move in toward the eye, all of the light rays get bent, and that focal point actually falls behind the eye. Now we can correct this by wearing glasses or having some type of convex lens that will move that focal point more forward and onto the retina. And if you are nearsighted, which I am as well, that means your eyeball is too long. So the light rays enter into the eye, and that focal point falls in the eyeball itself rather than on the retina where our photoreceptors are. So that could be corrected with a concave lens to focus the light rays right on the retina. And this is called myopia, and farsightedness is called hyperopia. And let's walk ourselves through visual pathways. You can see that our photoreceptor sensory neurons are in the retina in each of the eyes. So those photoreceptors have their axons move through the optic nerve, so you can see this on either side, and extend all the way to an area known as the optic chiasm. Now at the optic chiasm, we are going to have some of these axons stay on the same side, whereas the others are going to cross through the optic chiasm over to the other side of the brain. So once they cross or not cross in the optic chiasm, we create optic tracts. And these tracts also still have those axons moving through, and now we've got information from both eyes that are going to project over to the thalamus. Now over here we are going to see one of the nuclei within the thalamus. This is your lateral geniculate nucleus. This is important to know for your exam. The lateral geniculate nucleus is going to take the information from these axons, and we synapse onto other neurons that are then going to travel through what we call projection fibers, or sometimes radiation fibers, over to the occipital lobe. Now also notice that we are going to have some fibers make contact over here at the pretectal nucleus of the midbrain. We're going to have just a few fibers here that will synapse, and then we have our also synapsing over here at the superior canaculus of the midbrain. Going back to our pretectal nucleus, I want to let you know that this area is going to be responsible for mediating non-conscious behavioral responses to acute changes in light, which you don't really need to know for your exam. And then the superior colliculus is going to be responsible for integrating visual, auditory, and somatosensory spatial information to initiate orienting movements of the eyes and head toward salient objects within the space. that you are in.